we want to know what is above what you expect by chance. And there is firing rate, right? The neurons are firing with higher or lower firing rate. And the higher the rate of both neurons, the more you anyway get. So we want to account for that. And by this, you calculate the probability of firing in a bin by counting and then looking how much is there, so to say, in a bin and what probability is this. And if you multiply the probabilities for both neurons, this gives you the chance, the probability in case of independence.
The chance of synchronizing, let's say, a link of multiple neurons together is significantly close to a low chance when the amount of neurons.
No, when the number of synchronous events I measure is higher than the one I would expect given this probability. Yeah? Okay, and then, so now I say maybe, okay, maybe this is needed. Okay, and now I want to show you what kind of approaches we tried. I want to mention one more thing, which makes this really complicated in general, because analytical derivation of a null distribution is basically not possible. Yeah, you may want to assume Poisson, but if you look at these spike trains here, this is time, number of trials, 1 neuron. You see how regular this is. Yeah, it's more firing like a clock, and this is not this would not be a Poisson distribution anymore. So you need to use another one, and then it becomes really difficult already. And in addition, to consider aspects like a cross-trial non-stationarity, that you have different levels of base firing rate across the trials, it's also to be considered, or something like latency onset change, as you can see here, the neuron fires earlier, then here it starts later. All this needs to be considered in the null hypothesis, and we came to the conclusion that we need to do this differently than analytically. And therefore, I came across what we call surrogates, or is to manipulate our data such that they keep almost all the features of the data, but we destroy the temporal arrangement of the spikes. Yeah? For example, one way which is easy to understand, of each of the spike trains, you touch each individual spike and scramble it a little bit randomly. Yeah? Thereby you only touch, so to say, the exact timing. Or we explored a lot of different approaches, but then we ended up at this one. This is leave the spike train as it is. Don't touch the ISI distributions or any of this, but shift the spike trains randomly against each other within a certain time window. And this trial by trial. Thereby you don't touch the ISI, you don't touch the internal firing rates, but the temporal relationship. And if you want to look at the detailed analysis of the comparison of different of these surrogates, this would be Stella et al. Now, here you see just an example, even of different surrogates, showing that if this was done for coincidence synchronous spike events, so we had an empirical count, and we wanted to see how much I would expect by chance based on surrogates. And we used different surrogates like trial shuffle, spike randomization, or dithering of the individual spikes. And you see, even there, you get very different distributions. You need to do a good job that you don't destroy more than you want to destroy. So in some cases, it would be highly significant, in others not. And we, of course, explore this before we apply this to data. Now we want to turn to spatiotemporal spike patterns. Let me define this in a rather non-mathematical way. So again, neuron ID is time. And here I mark the pattern, this, different neurons. And we have a delta t between the first and the second spike, and the second and the third, and the third and the fourth. So these are the neuron IDs, and the delta t's between the individual spikes. And we search for patterns which exactly repeat by this, as this. We, as I said before, we look at a precision of 1 to 5 milliseconds. Typically, we allow a bin width of 5 milliseconds. But we request that the very same pattern and the same neuron IDs and the same timing repeats. Now, the first issue was, how do I find those? How do I count in, let's say, 100 parallel spike trains, these neurons which have such patterns? Yeah? And we were able to reformulate something which comes from market basket analysis. And we first translated this when we were searching for synchronous spike patterns. So which spikes fire together? And we used frequent item set mining to do this. This is just for counting. And there was a minor step then to translate the results of the frequent item set mining to do this for spatiotemporal patterns. This is efficient. There are efficient algorithms. And then if we want to evaluate the spike patterns, we do now the following thing. We have the original data. What we do with this data is we bin them in exclusive bins, as I said, 5 milliseconds. The content will be clipped to 0, 1, so this is binarizing. We apply frequent item set mining, then we have the counts of the different patterns. Then we destroy the correlation in the surrogate data, and this we do very, very often. And however, for the surrogate data, we also apply binning, clipping, FIM, pattern counts. Yeah, and thereby by this part, I have then, so to say, the expected number of patterns and can compare it to this. And recently, we extended a former analysis together with Günter Palm. Some of you may know him. He's a mathematics. Yeah. Excuse me?
I had common work with him years ago.
Ah, interesting, interesting.
I was in Ulm.
Ah, you were in Ulm. Yeah. So he did this together with us. So he defined very mathematically a quality of a pattern where we consider the total size, how many neurons are involved, how long is the pattern, how often did it repeat, what is the expected number in a more complicated way. And then we get for each individual pattern a quality measure. And we applied this now to the data I showed you already before. Because this is quite time consuming and because this surrogates and so on and going through the patterns, we do this in a quasi time resolved analysis by defining epochs around behavioral events of 500 milliseconds. So this is, the monkey initiates the trial, then there is a wake up signal, then there is a cue which tells him, later on, you may grip, you may move your arm and grip either with a side grip or a precision grip with different forces. But the information about the force he gets only later on. with the go signal, and then he gets reward. So we have for one session, which is about 15 seconds, we get about 24 data sets. Now, why am I doing this? I wanted to know, do precise spatiotemporal patterns occur at all? How many neurons from the total recorded neurons are involved in these patterns? And are they related to behavior? And what is the potential mechanism underlying the patterns? I started out with the Sonja chain as a hypothesis. And here you just see a first impression, I would say, that you see how this embeds. Just for you to have a look, you can see here the time, 500 milliseconds, different trials, only shown the neurons which participate in any of these patterns. And we inserted also, we show the patterns in color, which occurred here. And I will now follow with a certain statistics. Now, these are the different, what we call trial types: side grip high force, side grip low force, precision grip high force, precision grip low force. And we have these different epochs. And you can see here which patterns occur in these individual epochs and how often they occurred. And it's quite impressive that some of these patterns occur about, here in this case, 37 times in about the same number of trials. These are other trials. There are less, for example, in low force, less patterns. And in precision grip, there occur generally more patterns. They look a bit similar, but I cheated a little bit because they, per epoch, have the same color. And in low force, there are less patterns. We thought at a minimum number of occurrences, about a third of the trials. And we may have up to 45 occurrences of such a particular pattern.
Give us a sense of the time scale.
Yeah, timescales are here a bit. This is the experiment timescale. This is about to the go about 1.5 seconds. There is no timescale. This is just showing these patterns. Sorry, I also forgot the y-axis. That's true. So yeah, so one trial in total is about 2 seconds.
How long is one pattern?
They may have occurred up to 100 milliseconds, but they didn't. I show you in a minute. Up to 100 milliseconds, but they didn't. So now what we found is, it's a bit hard to see. Individual neurons participate in multiple epochs in different patterns. This is confusing for me, I admit this. I just wanted to illustrate this a little bit, how involved this all is. You see here by this is one pattern composed of these 3 neurons. All of these, so to say, balloons are one pattern, and they share individual neurons. And for example, this one is a highly shared one, and that one, and that one, in different patterns across different epochs. And this is not only in this trial type, but here you can see the different trial types. This I find particularly interesting, which appears a little bit as it would be a sequence, but still they share a lot these patterns. And this, of course, are neurons, and this is something which I hardly can imagine how this should occur from a Sonja chain. Here is just as a numbers, how many each neuron, here are the different neurons, which participated in a pattern within this one recording of 100 neurons. These are about a quarter of them. And individual neurons occur in different epochs, in different child types, in different individual patterns. This is amazing, I find, and I don't know how to explain this. I also don't know what kind of assembly this should be. Another aspect I wanted to show you, which I found also interesting, is you see here, so to say, the layout of a Utah array. And so we can localize the electrode at which a particular neuron is recorded from. And assume we have now a pattern from this electrode and this with these neurons. And we know their delta t, yeah? But as we have this array, which we know the size of, we can also calculate the distance in space between these individual neurons. And I wanted to know, is this a trivial effect, what we see? Is there, so to say, just a propagation or something, and we just see the delay? But here you see the scatter plot of the inter-spike times of the patterns and the inter-electrode distances. And this is the correlation coefficient, 0.096. So there is no correlation, yeah? So if there were, as people proposed, and we also thought about this, that there are waves of local field potentials also, you would see a more systematic aspect, but I don't see it. So this is what I can tell up to now. So individual spatiotemporal patterns of a precision of 5 milliseconds occur at least 10 times and up to 45. Way above chance, they are composed of at most up to 5 patterns, and the individual neurons occur in multiple patterns. Now I want to make a short jump, and we have also the expert here, Mihai. We had a collaboration. We have a collaboration with Mihai because I was wondering whether in AI networks, which have a relatively similar structure as Sonja chains, right? This group and group and group, this feed-forward structure. I was wondering what is happening is in such a network. If in a Sonja chain, you hardly can avoid to have this synchronous activity propagating, but what is happening here? Anu was also involved, sorry that I didn't mention you. So what they did is they presented these MNIST samples, handwritten digits, to, in this case, a 4-layer network with excitatory and inhibitory neurons. And what I found quite smart, because everybody told me, no, you cannot train a spiking network because you cannot make a derivative to derive the loss function. However, they said, okay, the right neuron should fire first, which is a smart idea. In addition, there was another aspect that an image like this, where you have darker pixel and whiter pixel and so on, was represented into the first layer as dark makes a first spike, and the whiter the later. These were leaky integrated fire neurons, and as I said, trained to report a time to first spike. Now, what is happening in this network? So if you look at individual digits as an input and you look at the activity of the neurons, first layer above are excitatory neurons in blue, inhibitory, excitatory, inhibitory, 4 layers, output neuron. Excuse me? The excitatory are red and the other ones are blue. Here you see the input and how, with which delay the input is arriving. And if you look at this, it looks very much alike, and you think, how should this thing differentiate between the individual digits? However, if you look at individual neurons in the individual layers, I come from neuroscience, I look at the tuning of these neurons. How do they respond to individual inputs? And you see they, at the beginning, no, not so clear to what they respond. Here in the next layer, it's this. And then the preference becomes rather strong to a particular input. So there is a tuning developing through this network. And here you see the measure we used this information gain. And here you see that at the very end, the neurons are highly tuned. Now, this is also, I think I can skip this, also the joint activity of the neurons through the layers becomes more and more specific. But what I found also interesting to then, we have in such a network, we can look at everything. This you can't do in neuroscience, right? Here we can not only look how they are active, but what connections they have learned. We can look at the weights, yeah? And what neurons, what Jonas then, who was the first author, thought he may look at an output neuron and see from which neurons of the former layer does it get strong synapses, yeah? For a particular stimulus, of course. And so he went back through the individual layers and defined a path to each individual output neuron. And then he looked how the, so to say, path membership developed through the layers. So here it's rather broad. An individual neuron is rather broadly participating. But then in the fourth layer, there are only neurons, so to say, participating in only a few of these paths to the correct output neuron. Now putting these links together, you know, functional connectivity and so on, activity and connectivity. If you now look at the digit, presented digit 4, to the path to output neuron 4. So we look only at these neurons which were indicated to be part of the path. You see that, and this is the red and the blue activity. You see that this synchronizes strongly and has a lot of activity shortly before the output neuron is being activated. If you have the same input to the neurons of path 9, you see that this activity is more or less not active, and only much later this neuron is being active. So, certain connectivity prefers a certain input activity. And at the very end, we observed that there's yet another aspect. You can see here, so to say, the population activity through the layers. And we define something like a pulse packet, as Markus Diesmann did, where we said, okay, to the highest response, we consider this as a spread of the spiking, this part, this spread, and how many neurons are active at the part of this peak. And now you can follow this through the layers. So you start here with, let's say, 40 neurons being active with a very tight activation. Then it gets widened and then synchronizes back again towards the end and becomes again a bit more neurons, similar to the inhibitory neurons. And this reminded me very much on this figure by Diesmann, Gewaltig Ärzten, 99 Nature, where they found that if you start such a synchronous packet and characterize it by the number of neurons being active and how wide it was, that if you are above a certain line here, you can stimulate with a certain number of neurons very tight, and it runs step by step into an attractor. This reminded me very much on this. However, we seem to come from somewhere above here and run into this attractor for the individual digits. Now, in principle, yes. If you stimulate the right neurons, this is going in this path. Yeah? He was able to do this for this.
So later, when Würdecke and others derive analytical conditions for why this synchronization happens and the attractor exists, but still there is no full analytical theory that describes the whole process. Somehow very hard. The synchronization condition we have analytic, but not a full description of the dynamics.
Thanks. So a last slide before the conclusion. We also looked at the overlap of the path neurons and active neurons for different sample presentations as a function of the layers. And here you see that the path neurons deviate strongly from the ones which are generally active, meaning, now comes the interpretation, that we saw before that the overlap is decreasing a lot. So this must be paths which at some point don't have overlapping neurons anymore. Yeah? Or in a picture, we have an input layer and we have these individual layers, but the effective path to one output neuron 4 or 9 separate at some point. They don't have an overlap anymore. Yeah, this is why we made all these computations. And this is a figure from Moshe Abel's book from 1892, which he had in the discussion section in his book, which is exactly this. He predicted this many, many years ago, that something like this is possible, that you have overlapping chains in principle. Yeah, the one goes there and the other one goes there. Okay, now I'm at the end. Massively parallel spike data are a challenge for the analysis of temporal correlations. Surrogate data help to implement the null hypothesis. Potential signatures of thin-fire chains assemblies were found by above chance synchronous activity at behaviorally relevant times. And spatial temporal patterns, spike patterns, occurred way above chance and are specific to behavioral context. And all this occurs despite the massive subsampling. And deep spiking networks express thin-fire chains for classification of MNIST. And by this, I want to thank the people who participated here and contributed to the various aspects. Cristiano Köhler, Junji Ito, Jonas Oberste Frielinghaus, Arno Kurt, Günter Palm, Tobias, and so on. And the externals are not shown here. Thank you very much.
Sonja, I think we have a few questions and then we can copy on that. Nobody, I want to ask about these electrodes. Are they, how they are positioned? Do they report from single neurons? Is there a guarantee that each electrode is, doesn't move during the thread?
Yeah. So we did not analyze the record. This is chronic recordings. We don't put this in a pot with different recording days, but the assumption is that the neurons stay constant within a day. Yeah. Of course, when the monkey is then outside in the cage and so on, he's moving the head and so on, then everything and so on. Yeah. But we assume that they go into the cortex. Huh? They are probably in layer 3. Mihai? This was motor cortex. Arno?
You said earlier, like that were puzzled by single neurons participating in data, right? So I think this you can also parallel to this alternative theory of how the cortex works via the latent factors. We also find that individual neurons contribute to many factors there as well. So it's a general theory and a potential kind of connection.
Yeah, but coming from, so to say, this feed-forward network, yeah, it didn't fit. Only if an individual neuron is participating in individual groups later, yeah. But this is so intermingled. I'm not sure if this would work. However, you want to continue? Somebody in our lab, Tom Tetz's lab, was doing modeling of how sequences could be embedded in a network. And he was, so to say, using, how should I explain this? That an individual neuron can be embedded in, no, an individual neuron standing for a certain digit or letter can be embedded in different sequences by activating different synapses on its dendrite. Because there you have such a huge combinatorial option, yeah, to embed this. And so in that sense, it could fit again, that an individual neuron is really embedded in different constellations, but getting from somebody else the input. Yeah.
And this connects to what I wanted to say. I mean, you say, I agree, like to fit perfectly to the forward view, but of course, the motor cortex is not forward.
Yeah. We need to discuss a bit more about this, yeah.
Well, I mean, this goes into the same direction that my question was. So, thinking of the distance between the neurons, I assume we're talking millimeters here, right? The entire side. And the time separation that you mentioned was on a scale of tens of milliseconds, is that correct? Right? Now, thinking of what typical delays could be, this sounds like more than one synapse, right? 30 milliseconds. So I think it's unlikely that, you know, if you have 3 neurons in a sequence, that neuron 1 actually activates neuron 2 and 3. It's rather that they either go by other areas of cortex or they are co-activated by something else. Which is very different from the visual cortex thing that you showed at the end with the digit classification.
Mm.
Just a completely unrelated question from a medical point of view. If you had delay for each usual factors as a career, like bleeding, like scar, like medical structure, that is delaying the.
Yeah, but repeatedly in the same.
Talking about the same areas and they.
No, this was one area.
Yes, then basically the career would be there or remain there. Or
I don't think that this is the cause, but I cannot exclude it.